Hello, my name is James Hansford. I'm a postdoctoral research fellow at Northern Illinois University and the Zoological Society of London's Institute of Zoology. I've been researching megafauna in Madagascar for a few years, and my interest is in how they're now missing from Madagascar and how we can understand their ecological roles and the relevancy to modern ecosystems. Um, So I've been studying elephant birds in particular for about 8 years, and I've been putting in some research about the underlying taxonomy, extinction timing, and currently their ecology through isotope analyses. I'm also working on projects with their crania as well to understand more diversity and ecosystem function. I have some skills in multivariate analyses, niche distribution modeling, amongst others. And my focus really is about historical extinction and how we can see that there are missing ecological functions and roles in Madagascar, and to what extent are they replaced by introduced species and those species that remain alive today. So I'm sure you're familiar with all of these papers, but during my research, I've been interested in a diverse array of habitats in Madagascar, and that includes the potential of grassland assemblages and those species that may have regulated them as through a grazing guild. In past research, people have focused on the roles of hippopotami and tortoises, but have often excluded elephant birds because they have had little underpinning research until the last few years. So when we break down isotopic niche space of megaherbivores at the species level, we can look at all of those elephant bird taxa, as well as the two recognized hippopotami taxa, Lemele and Madagascarensis. And we can compare that to what we might expect in carbon isotopes for finding a C4 grazing guild. So when we look at that, some species stray into that mostly C4 territory, but are predominantly down in the C3 and purely C3 and C3 dominated habitats and roles. Whereas we have the elephant bird species Epionis hildebranti, which seems to stand out as a narrow banded niche that may have been indicative of grassland ecosystems. With their species average level and being restricted to the Central Highlands, they may have consumed 40 to 50% of their diet as seafood grasses. We do need to get some better underpinning plant data for isotopes in the region and across Madagascar as well, as we're using global averages of C4 plants to make this estimate at the moment, and we'd like to do better. So we can see that whilst Epionis hildebranti is probably the standout as that narrow-banded C3, C4 mixed guild, we can see that hippos also delve into that territory as well. And we are missing data on tortoises from the Central Highlands region. When you split it up on a taxonomic basis, so we have one species, Hippopotamus madagascarensis, which occurs in the Central Highlands and outside. We can see that outside they have the signature you might think and associate with C4 grasses, whereas in the Central Highlands they don't. This suggests they might be switching to they might be CAM specialists, which switch their signature in wet and dry conditions associated with these habitats. When we look at all hippo data across the Central Highlands, though, we have a slightly different picture. So in Ampasampa and then the two higher elevation sites of Saramodi and Ansura Bay, we get quite a wide variability in isotopic signatures, suggesting that there is some variation associated perhaps with habitat fluctuations through time, which we need to draw out a bit more. So my own research limitations are numerous, I might say. Carbon isotopes can create false positives, which may lead us to interpreting grasslands that should actually be sedges and rushes. Nitrogen isotopes can mitigate this, but we need some expert support through people like Brooke Crowley, and we can need to develop grass isotopes from varying sites across the Central Highlands as well, which I know people are working on, and I'm very excited to look at that data at some point. Population size, if we do have a species such as hildebranti, regulating grassland in Madagascar throughout the Holocene, we need to understand not only their distribution, but their population size as well, to see if they could have regulated grassland on a large extent. But ancient DNA methods have so far failed when I've been working with people. Do we keep trying again, or do we wait for technology to get better? We don't know. Temporal resolution. So most of my data are limited to the mid to late Holocene, and investigating across more of a range of time would be useful to understand fluctuation and change. And understanding to what range extent these species exist. So are they only at high elevation sites, or do they have topographical limits where they need large flat areas for grassland ecosystems? And we need to develop more excavations, material and context for this. So I say just limited distribution. So currently there are quite a few sites in the Central Highland region which yield megafauna, and Apionis hildebrandt only comes from 3 quite closely associated sites of Cerramodi, Ansara Bay and Masalandrona, which are all quite high elevation. Whereas you go to slightly north to Ambasambasimba, we have no Apionis hildebrandt whatsoever, and it's quite a different guild of animals there. In the future, I'm looking at going beyond bivariate analyses and thinking about trait probability density. So you can use variable traits, quantified traits, such as isotopes, in trait probability densities. So we can use that to quantify functions of richness, evenness, divergence, redundancy, and replacement, which are quite important for my research. But we can also include other data, such as body mass, so we can compare large against small species, dispersal, strontium isotope variability, perhaps, or sulfur isotope variability, hydrological associations with hydrogen isotopes, body data from leg morphology, etc. These all things are possible, but to compare on a broad range of taxa, we need to have something that's comparable against them all. And temporal association, observing functional change in response to other variables that can come from, hopefully, from other people in this workshop community. Variables such as climate, pollen records, fire records, all of these things can be linked together, and we can understand how ecosystems and animals change and respond to different conditions over time. So that's it. What I want to know is, what extent are animals in Madagascar today that have been introduced by humans replacing what we have lost?